University of Central Punjab, Lahore, Pakistan. I am working as an associate professor and head of the Department of Biochemistry. I would like to extend my thanks to BioSolveIT for giving me an opportunity to present my work over here. So without further delay, I would like to start my topic at unveiling potential of DNA polymerase 3 subunit beta inhibitors to limit the H. pylori, Helicobacter pylori. So, as we all know, H. pylori is a gram-negative spiral-shaped and flagellated bacterium. It actually infects about half of the world's population by mainly causing the chronic gastritis and peptic ulcer disease. It also has the determined pathogenic role in the development of distal gastritis, adenocarcinoma, and gastric mucosal associated lymphoid tissue lymphoma. In fact, it is also a contributing factor for the gastric carcinogenesis by stimulating the gastric cells proliferation without counterbalancing with the adequate apoptosis. The treatment of H. pylori infections has changed drastically in the recent years due to the increasing antimicrobial resistance. Therefore, new drug target has to be identified to overcome the resistance mechanism and to treat the H. pylori infection. So, coming up with the next. Taking into account the transmission of the H. pylori, if we can see the slide, it usually enters through the oral route and swims rapidly through the viscous mucus as it possesses the unipolar flagella. This characteristics allows it to move from one location to another within the stomach and to more favorable areas where required. In this image, actually, what is shown? The mechanism that H. pylori secretes large amount of urease, which effectively break down the endogenous urea to ammonia. It not only produces or break down the endogenous urea to ammonia, it also creates a favorable condition for the survival by increasing the amount of ammonia, thus causing the pH level in the environment to rise, and it protects it from the acidic nature of the stomach environment. Urease can also have some toxic effects on the cells of gastric epithelia. As such, it contributes to the infection and stimulates the infection and immune response. Now come up with the survival for the H. pylori. Besides the gastric diseases, H. pylori can also affect the other parts of the body and negatively associated with infections. If you can see this slide, the green ones and the yellow ones are actually showing the correlation. That means the diseases in the green boxes are caused by the H. pylori, while the mean in the yellow disease squares, these are actually inversely correlated mean with the presence of H. pylori. That is, H. pylori is beneficial for these diseases like asthma, inflammatory bowel disease, and the celiac disease. H. pylori actually causes a ratio of the T helper cells, Th1 and 2 imbalance, which can trigger the multiple sclerosis. Moreover, induction of the interleukin 6 and 8 and interferons by the H. pylori causes the periodontitis. Similarly, H. pylori can also cause the ischemic heart diseases due to the increased risk of T helper responsive atherosclerosis. It also triggers the activation of TNF, NF-kappa B pathways and most of the chronic inflammation, again elevating the level of the C-reactive proteins. Due to this, TNF-alpha and interleukin-6 insulin resistance increase and type 2 diabetes mellitus is caused. Moreover, bacteria utilizes the iron from the host and it results in the redistribution of the transferrin, hence causes the anemia of the patients. While with other things that H. pylori is beneficial for the host in the way that it can also induce the TREG markers, that is CD3 and 4 markers, that help in the reduction of the risk of celiac diseases and again with the childhood asthma. Again, the induction of the IL-10 and suppression of the T helper 17 cells due to the infection of H. pylori also limits the development of inflammatory bowel diseases. Coming with the next figure, actually this figure attributes to the that antibiotic resistance in the H. pylori. You can see there number 1, 2, 3, 4, up to 7 resistance mechanism. That is, if we can start with the first, that is macrolide or rifamycin resistance, how H. pylori resistance mechanisms are there. Number 1, structural mutation of the DNA gyrase subunit A and B, that is DNA-dependent RNA polymerase protein, induces a reduced affinity for the fluoroquinones or rifamycins. And this helps protect the bacteria from the DNA synthesis and obviously transcription from the antibiotic effect. So this is how the H. pylori resistance comes. Second mechanism, if you can see the resistance of mutation of the 23R RNA genes and those of the 16R RNA genes that prevents the macrolides and tetracyclines. Here I have quoted the examples of clarithromycins and tetracyclines. And these act on the ribosomal 50S and 30S subunits respectively. The mutation therefore protects the protein synthesis from the antibiotic effect and allow the survival of the bacteria despite their high intracellular drug concentration. Coming up with the third penicillin binding protein, that is penicillin binding protein A1A, is altered due to the structural mutation that displays actually a reduced affinity for the binding of amoxicillin molecule. Consequently, this cross-linking of the peptides during the last stages of the peptidoglycan synthesis in the cell wall is prevented, and this inhibition causes the antibiotic effect. Prevented the antibiotic effect. Now the diagram that is labeled as 4. Actually, it is representing the drug activation failure within the H. pylori. How? In this part, mutation that will altering the function of NAD-dependent flavin nitroreductases and oxygen-insensitive NADPH. Oxygen NADPH nitroreductases actually suppresses the metronidazole enzymatic reduction. This enzymatic reduction leads to intracellular accumulation of inactive product, and after this inactive prodrug activation, the upregulation mutation causes the increased activity of the DNA recombinase A. Therefore, DNA repair system are superoxide dismutase effectors, and it induces the regeneration of inactive metronidazoles and protect the bacterial DNA from alteration by metronidazole toxic drugs. Next, you can see the biofilm formation. If we can see there, although it exists but as yet limited, or hypothetically we can say that clinical relevance, it consists of the matrix of polysaccharides that actually constitute of multifactorial barrier in the drug penetration and drug activity. Now, next is the reduced uptake, drug uptake, and increased drug efflux mechanism that is mediated by the upregulation or overexpression to the efflux system. The pump system and downregulation of the outer membrane protein that reduces actually the intracellular concentration, which below the lethal doses as it is represented over there. Coming up with the 7th last, that is the formation of the cohorts that probably generates the ultrastructural and metabolic changes that are able to prevent cellular penetration and the activity of antibiotic molecules. So all these reported mechanisms are not working well. That's why, owing to these resistance mechanisms, a new drug target was essentially required. Now, why we have selected the drug target as a DNA polymerase 3 subunit beta? These subunit beta, if you can see here, they consist of chain 1A and chain B. And both of these chains have actually 3 domains. It encircles the DNA with the help of the clamp loader in an ATP-dependent manner and slides along with the DNA. And because of its ability to slide along the DNA, the clamping clamp is required by many different enzymes for DNA replication and repair. That's why the protein is essential and important. Moreover, its molecular function for the same protein are that it performs the numerous roles in the H. pylori and has hydrolysis activity, exonuclease activity from 3 to 5 prime, then catalase activity along with the nucleotidyl transferase, organic binding compound molecule binding, and DNA-directed DNA replicating. Now, if you can see that this perform the main quality of the protein is the interaction that this protein is showing with other proteins in the host molecule, that is H. pylori. Actually, if you can see that image, DNA polymerase beta 3 subunit, also known as DnaN. This DnaN is actually interacting with all the other several proteins. Like DNA X, if you can see this over here, it is also known as DNA protein polymerase 3 subunit gamma. Like R is beta, it is interacting with the gamma that is responsible for most of the replicative synthesis. Then DNA E, if you can see this, that is DNA polymerase 3. That is actually a complex multi-chain enzyme that is responsible for the replicative synthesis in the bacteria. Then come up with the pol A, that is in addition to the polymerase activity, it also has it exhibits the exonuclease activity from 3 to 5 and 5 to 3 prime. Coming up with the gyrase G, that is here, gyrase B, that is function as the type 2 topoisomerase. Another one important DNA A is the chromosomal replication initiation protein, which plays important role in the initiation and the regulation of the chromosomal replication. And it also binds to the origin of replication. I hope it's clear now, because these all are the mandatory steps that we want to include in the study later. Now, if we can see that how we design the best lead molecule using the BioSolveIT softwares. Actually, to my knowledge, to me, is BioSolveIT softwares are ideal because they give results swiftly with the precision. Actually, the infiniSee and SeeSAR are the main tools of BioSolveIT, while others are actually inbuilt in them. For this project, first of all, we have used infiniSee for searching the similar compounds from inbuilt library chemical spaces. Then we have used the CoLibri and the FTrees to determine the extent of similarity between query and the subjects. Subsequently to this, virtual analysis was carried out by the SeeSAR, and the best hits were structurally modified to improve the affinities by ReCore to obtain the desired inhibitor. Now let's have a brief introduction about the role of each softwares this we have utilized in this project. Starting my discussion with the infiniSee. infiniSee is actually a chemical space navigation platform that screens the billions of compounds to find the similar compounds to the query molecule. So it has a total of 5 inbuilt chemical spaces. The foremost purpose of these spaces is to provide the analogs with R synthetic or required access. First of all, chemical space is the freedom space by ChemSpace. It contains all the organic compounds that are based on the chemical reaction and can either be ordered either for our own, either the final synthesized products, or can be in the form of building blocks that for us is convenient and independent synthesis we can do in our labs. Second one, we have the ChemRia, that's Ottawa's on-demand chemical space. You can see that it further expands the searches horizon of infiniSee by allowing the search in the new areas of tangible molecules. Then third one we have is in its partnership with the Anamin Limited. It contains the world's largest collection of commercially available compounds. Using this CoLibri, the database contains the Anamin synthesis protocols and in-stock starting materials as a searchable chemical spaces. Real spaces allows the efficient hit exploration, from finding previously unknown analog to the scaffold hopping. Then fourth one is the Galaxy chemical spaces, which has been formed in close collaboration with the ReCore lab network. This space contains a lot of unique chemical intellectual properties with the research, and the compounds can be created on demand upon request to the ReCore. Then we have the last chemical space in the literature-based virtual chemical space that covers the thousands of billions of virtual products with a high likelihood synthetic accessibility. Then a short introduction with the CoLibri and FTree, how we have explored them in our project. These are two important tools in infiniSee. That is the first CoLibri, that is actually toolkit for the chemical space navigation that transform the synthetic know-how into a chemical space. Second important software is the FTree for pharmacophore style similarity searching, and actually we have done both, so we use that. It aligns the hit with the query and predicts the similar between what is our subject and the query. The similarity of such two descriptors is based on the alignment that is represented by different colors. And the coloring of substructures help us to identify where substructure of the query is matched onto which of the hit molecule. This is actually depicted by the score ranging from 0 to 1, where 0 is dissimilar and 1 is identical. Like binary functions, either a 0 or a 1. Then after infiniSee, that comes the SeeSAR. That is actually a visual drug designing platform. It has several features as it is used to explore the protein structure as well as the molecular modeling and also in the improvement of the ligand receptor affinities. Its most and utmost feature is the hide, which actually visualizes the affinity of ligand in the binding pocket and calculates the realistic energies of binding, and also pinpoints the affinity problems of the bound ligand. Also, it tells about the desolvation penalties, weakness of the hydrogen bonding, or indifferent scaffolds. Then we have the flux docking. That actually places a ligand in the binding pocket and split the ligand into the fragments. And these fragments are then multiple places in the pockets, and it uses the ligand-based or the protein-based pharmacophore that only constrain the post-filter as such to find the wide solution spaces. Then another important feature, that is the ReCore. To me, it is most fascinating tool, BioSolveIT software, because it required actually the 3D fragment libraries to search in, which is called the index. Through PDB, zinc, or CSDs are used to create its indices. It works on the shredding and the filter rule. The required fragments are kept by the shredding rule, and unfavored or unrequired fragments are actually excluded by the filter rule. It can generate also the new scaffolds in 3D rapidly. Last vital feature in the SeeSAR is ADME analysis. That can actually important factor to decide if the compound is promising drug candidate or no. So early assessment of the ADME parameter can predict the toxicity issues like absorption challenges are in the drug discovery and saving the time and resources, obviously. It can obviously calculate the rotatable bonds, TPSA, log P, or log S, etc. Now, if we can start how we have started the 2D searching similarity, then we have firstly downloaded the inhibitors of the protein, and then from the PubChem databases, then we downloaded various inhibitors based on their ability to restrict the protein or the activity of the protein. In this case, inhibitors are derived from the literature. Firstly, we open the infiniSee. It can show the plain screen. The top bar shows the several icons that are used in the process of 2D similarity. In this case, click on the icon of the selected libraries to select the libraries to perform 2D similarity. The option of the adjust parameters will help us to adjusting the minimum similarity and the target similarity too. Now we have clicked on the icon, that is file icon, and then click on the load molecules. We have loaded one inhibitor and selected the play icon for the 2D similarity. After some time, it will provide the result from the selected libraries and the number of the similar structures actually that are extracted. We have extracted these, and these were all from those libraries we have extracted, selected databases. Then we have clicked on the file icon, and this project was saved, either in the form of this file, or we can also export it in the SeeSAR file directly. And these were the parameters: target similarity, total diversity, minimum similarity, or the number of the results were obtained. 100 per search we have obtained these. Then after this, the process of this was obviously for all the inhibitors, repeated for all. After performing the 2D similarities, we opened the SeeSAR for the further procedure. These were actually overall searching the molecules. Then we have started open the SeeSAR for the further procedure. In this step, the protein domain was selected. Okay, so in this step, the protein domain that is selected was the protein. Select the protein from the PDB ID that in our case was 5G4Q. We open the SeeSAR and then we have written the PDB ID to that protein that is 5G4Q. If you can see my screen, it is written here. Then we have selected the protein and click the green button. This, if you can see this green button over here, we have applied this icon and the protein is ready for the docking. From the top down tab bar, we can add or remove the water molecules, amino acid, and etc. and adjust the proteins. With removing the water, are chosen the amino acid chain, B chain. We have selected actually the B chain. Then click on the protein and add it to the molecule docking mode. This was selected B chain and it was selected added to the binding mode. It is important to load all the 2D similarity searches and we have selected it, added it to the binding mode, and it's important to search for all the binding modes. In the docking mode, we have selected a protein as a reference and then we have changed the color. We can also change the color. It will upload all the inhibitors and the analogs to our desired protein. Then we perform the docking and click on the green button. Started the standard docking to generate the poses, and after some time, all the poses were generated. Then to check the, if you can see there, then click on the check of all option and calculate the affinity, hydrogen bonding, torsion qualities like optimum properties and some of the clashes also. The best affinity molecule and the torsion qualities are virtually screened through this process, and then compound that was showing the best results were then selected. After some time, all the affinities are actually showing over here. If you can see this, we have checked all through this and then estimated qualities, torsion qualities, clashes, and so on. So this is working, it's on. Then we have, after some time, the affinities have appeared over here, estimated affinities. Then we extracted the poses with the best affinity. You can check the best binding in the analyzer mode as well. All these selected compounds actually showed that the best binding with the protein and were processed further. For our result, we have extracted 56 poses from 2,212 poses. So the 56 which shows the best result depending on our selected parameters. And the next step is to create the pharmacophore. So how we have created the pharmacophore, actually it is possible that selecting all the poses of the protein at the same time, all the protein and poses one time helps that generating the best pharmacophore. So we did this. If you can see my screen, and all the inhibitors were overlapped on the protein, and that helped determining the best hit. We created a pharmacophore based on the binding between poses and proteins. This created the pharmacophore that click on option to define pharmacophore, and then we adjusted the pharmacophore. Click on the option of the define pharmacophore, define new constraints, and it also adjusted according to the structure. Once the pharmacophore is created, all the possible interaction between inhibitor and protein is visible, if you can see there. After this, we shifted these poses to the Inspirator mode, where it has the ReCore. Actually, this ReCore included all the databases of the zinc and PDB, and it gave us the similar structures. Moreover, also ReCore, that is in the Inspirator mode, it actually helps the replacement of the central elements, that is, we can say bioactive molecules, and generate the new scaffolds in the 3D, in the blink of an eye. Select one structure, like we have selected the yellow one, this one, and click the green icon, and then it will further allow us to select the particular area. so that different databases can provide the similar structure. And once we have added to Inspirator, you can't believe we have obtained total of 478 molecules against 56 poses. Initially, we got at 56. Then we have clicked on the check all option, like, for example, affinity, hydrogen bonding, torsion qualities, optimum properties, and then determine the clashes and so on. Upon which, if you can see this, we have structured the ReCore. So we have extracted the molecule with the best affinity and also determined their admin property. We can go with the analyzer mode, and then we check the binding of these inhibitors with the protein separately, one by one. In this way, our best inhibitor with the maximum capacity was selected. So if you can see this, actually these are giving us the numbers that I have mentioned before previously with this, and then these all the properties were calculated depending upon what they are giving us and why these are excluded. So you can see this, we selected this way and we calculated the admin property in this way. Now with this, we have obtained 3 of these molecules. Now, these 3 lead molecules we have received at the end of our whole work. We have synthesized 2 out of them, and then we are in the step of determining their in vitro and in vivo inhibition analysis. For example, in vitro analysis are the studies we have selected 2 types of urease enzymes to check them against them. Firstly, as the extracted that is commercially available as Jackmin urease, and the second one that we are in the process of extraction from the basically H. pylori samples. We are isolating and culturing that, and we will soon obtain that H. pylori, and we will try to check the inhibition of these compounds against those 2 H. pylori and Jackmin urease inhibitors. So the concluding remarks may be that is possible to overcome the resistance mechanism of H. pylori by targeting novel druggable molecule protein. DnaN can interact with a number of cellular vital proteins, so inhibition can alter the overall cellular functions. Thus, screening of inbuilt chemical spaces in the infiniSee provide the suitable druggable molecule. The best poses of the drugging analysis were actually modified and lead inhibitors with the highest affinity after the admin were obtained. And at the end, I would like to thank my whole group, especially these 2 who has carried out this whole project under my supervision.